And welcome back to the Atma paradigm. Our title today is What's Life? And we'll explore that from the standard story offered to you in schools and universities. But we might give a different perspective on it. Specifically today we're looking at theories as to how the first living entity that might have been came to be, how it came to be assembled from simple chemical components. Now this process of self-assembly for the first living thing is called abiogenesis. So today's title, What's Life? Is the analysis of abiogenesis, and it's a critique of prebiotic self-assembly. It's a big topic, and there's so many competing theories and ideas which really don't all thread together. I can't cover everything, but I will focus on a batch of the key issues, and particularly as they pertain to the Atma paradigm. So, and what we'll see from all of this is that this analysis of life and where it comes from doesn't really help us understand the nature of life, nor does it tell us very much where it comes from. But first, a short recap from where we were last week. What we covered so far was, and this also sets the intention for this set of talks in part 3, consciousness is a primary aspect of reality. Consciousness interacts causally with matter. Those are our primary points that we have established, and we will use those to look at the rest of the big questions of existence. This interaction of consciousness with chemistry produces the special life functions that we call biology. Now, there is no accepted theory for the biological origin of life, as we shall see. And in the next couple of talks, we'll look at the evidence cited for evolutionary theory and shows how that reveals a different picture of life's origins. So from all of that, having established those points, we will then, in the later talks of this part 3, propose a new consciousness-based system for the appearance, proliferation, and the adaptation of all species. And this is a consciousness-based view of life. It shows its origins, its proliferation, its adaptability, and it's radically different to the rather limited views of both evolutionary and creationist ideas. And this view of life does more than just explain the science at work. It offers a very different vision of all life, where we fit in. So it gives a new vision of ourselves in relationship to the living world. And it connects us and unites us as conscious beings, as the Atmas, with the life of all other creatures, plants, and anything else that's alive in this world. And it also reveals the incredible way the world works so as to provide practically unlimited opportunities for sensory experience and enjoyment within all species. So I hope you're going to stay with us for the next few talks. I hope that they're interesting and exciting, and that you'll enjoy them. Now, we're going to start with the key issue of the physicalist narrative. What is at the heart of the debate on issues particularly of life? And it's the problem of information. The big problem for science and philosophy is to solve the riddle of why there is so much information inherent and embedded within all the specific forms that fill our universe. Everything within the universe contains information, but not just simple information, complex specifying information, as we have defined in previous discussions on that. Specifying information that defines the structure, form and functionality of all the things. The troublesome issue for science and philosophy is, where does this specifying information come from? And we're going to be looking at that first in terms of life, particularly what information does it require to define a single-celled organism, that first simplest life form of all that is talked about. And then next week we'll consider information within living systems. So what is life? Well, according to the BBC website, there are over 100 definitions for life, and they're all wrong. And that's understandable, because it's a really big challenge to define life for science. Because life is a process, not a substance in their mind. And you can't say it's this chemical or that chemical. There doesn't seem to be one specific thing in the physical world that you can point to that says this is life. So any definition has to be broad. It has to encompass all the different forms of life that we know on Earth, and possibly any that we might find or come upon anywhere else in the universe. So here's one of the better efforts for a definition of life based on the kind of biological approach. It says that condition that distinguishes animals and plants from inorganic matter. Now, okay, what distinguishes inorganic matter from a living thing? Well, they're different, okay? So we're not clear on what this distinguishing feature is, only that it includes the capacity for growth, reproduction, and functional activity. So we're saying those three things shouldn't really happen within a non-living thing. Still doesn't tell us what life is. It's just that now we can say we know a few things about what life does. Growth, reproduction, functional activity. Oh, and one more, and continual change. Now, why do they stress and continual change? It can't mean that life is changing from life to not life, because then that wouldn't be a definition of life. It's saying that continual change exists during life. And that's important, I think, because obviously the body is growing and changing as it starts from very small stage and gets into maturity and then ultimately dwindles and disappears. So, but it also, they include this because they want the opportunity for adaptation. Now, adaptation, of course, is the keystone of evolutionary theory. Once you get a living thing, it can adapt, and they say that produces evolution of the species. We're going to explore that more next week, but we don't disagree that there can be some little changes. And finally, all of this is happening preceding death. Now, this makes it a little kind of circular, that life is that thing that happens before death. So really, here's our definition. Life is the thing that living things do before they die. You know, although, you know, there's books and films about things to do before you die, a bucket list. So life is the thing that living things do before they die, and that kind of sums it up. So this gives us a definition for life. Not what it is or what causes it, but what it's like and what it does. Now, obviously, I would suggest these biological definitions are limited at best, and limited in scope and value. They tell us about what life might do in terms of how it acts, how it behaves, but it doesn't really get to the grips of what is causing life to be in that state. And particularly, they give no account or acknowledgement of consciousness. Now, that's not surprising, because the physicalist kind of narrative struggles to accommodate consciousness, as we have analysed previously. So no wonder they don't try and bring it into biology, because currently there is no theory that links biology with the production of consciousness. So therefore it can't be brought into a biological definition of life. But that's why we in the Atma paradigm feel the need to establish a clear model for consciousness and how it interacts with the body, the brain, and the senses, as we have been trying to do, and use that as a better framework for defining the nature of life. So any definition that leaves out the presence and potency of consciousness will struggle to explain how living organisms, and particularly the chemistry of living organisms, behaves in the way that they do. Because chemicals do strange things when they're associated with life. Here's a little chart. Chemicals like simple states. They want to be in the simplest energy state for their molecules. Whereas in living things, we see very highly complex chemical molecules and systems. and they seem to thrive on this and basically somehow or other maintain all of this. So chemicals just follow the simple laws of thermodynamics, get to the simple energy state, fine. Living things somehow or other seem to be borrowing on energy from elsewhere in order to hold the complexity together. So in many ways, they appear to defy thermodynamics. They do that by borrowing energy from outside of themselves. Now, chemicals naturally break down. And we know that when life leaves the body, the chemistry, which was so complex, so intricate before, starts to break down. And all these wonderful molecules that we have in our body start to become just simple gases and water. Whereas in living things, there is a constant process to maintain that structure, but also to grow it and even to reproduce it, which is something chemicals cannot do. DNA does not reproduce itself. You can take the chemical DNA and play with it on its own, but it will not do anything on its own. It does not reproduce. However, when it is in a living situation, something else happens, and that chemical molecule starts to behave in a very different way. So the other one that they recognize, and this is a kind of nod to consciousness, the chemicals are inert and living things are responsive. We know that living things are more than just responsive. They're not just automatons. You press a button and something happens, but there's sentience there. That's the other element. Now, in our analysis of life, we need to look at it in two phases. This week, we're going to focus on abiogenesis, prebiotic chemistry. This is the prebiotic situation, what happened in chemistry before any living entities or beings existed on this planet. Now, so prebiotic chemistry is associated with simple chemicals and how they come together through a process of self-assembly to form the first functioning living cell. Now, once you've got a living cell, then you can have the study of biology, and then you can look at speciation. You know, how come there's different life forms, and you can look at the issues that people describe through evolutionary processes, and we'll be starting that next week. Sometimes both these stages, prebiotic and speciation, are discussed as evolution, but I think that's inappropriate because the mechanism for each stage is different. For instance, the standard account of speciation by evolution, see our picture of a friend, our tiger friend here, has two factors. One is chance mutation shown by the dice, and the other is natural selection. So that's the standard account of how speciation happens. However, natural selection is not a process that can be applied to chemicals. They have no interest in reproducing themselves for any higher purpose or for survival. So during the process of self-assembly of chemicals, before there is any life form, you can only rely on the chance interaction of simple chemicals and the ability of those chemicals and their properties to interact with one another. Now, we'll talk about this prebiotic stage and see how far this kind of restricted causality of chance can get us. Now, we just need to take a moment to remind ourselves of something we dealt with way back in talk number 1 out of part 1, and that is that we don't regard these particular factors as being scientific explanations or being very useful even philosophically. Chance, randomness, chaos, whatever you want to say, these suggest there is no cause. If something has no cause, it cannot be, by definition it can't be studied to satisfaction within the scientific process. At best, it remains a vague philosophical premise, but without ever hope of empirical verification. And we'll be hearing a lot about chance and random interactions in this particular session. And we mentioned about infinity or eternity by saying that something has an infinite number of chance opportunities, or something will eternally happen, then eventually we will get a particular outcome. So trying to link chance and infinity or eternity is, again, you may consider that a conjecture, but it is not science. It will never be tested scientifically. Then out of nothing and magical emergence. These are topics that we talked about previously. They're not so important for today. We shall move on. But we'll be watching out for chance, and we'll watch out for infinity to rear their heads during this discussion. We start with this idea that you can self-assemble building blocks according to their chance interactions. But building blocks are constrained by their own inherent characteristics. And we use the example of Lego bricks. Okay? Lego bricks, you all know them, have these little things at the lumps at the top, and they're able to fit together in particular ways. You know, they have inherent properties and they fit together. But there is very limited information within each of those units, and therefore the number of ways they can interact together is actually quite limited. Now, here's an example of kind of demonstration of self-assembly of the tobacco mosaic virus. It has a particular form, shape, this kind of cheese-shaped thing that you see at the top of the diagram. And it therefore kind of builds together in the way that's shown. And they form into these bigger wedges. And when these bigger wedges come together, they start to form either discs, which you see on the left, or they can start, those discs can overlap a little bit, in which case it starts to build a helical spiral cylinder. So this is actually very impressive. And this shows self-assembly without any help from outside, all based on the properties that the molecules have for themselves. That's great. Okay? Very impressive. They can do that, but they can't do this. By combining many small building blocks, you can have the ability to form so many different connections. However, something which is of complex structure cannot be said to have come together from those inherent parts, unless it's really by big chance. So the information for the overall structure is not contained in the individual blocks. It is not defined by the way they can interact together. It seems to need to have come from outside, or it's come by chance, some random interactions. So now this is interesting because, as we said before, chemicals don't want to be in these very complex structures. Even Lego bricks don't. They much prefer to be like this, scattered on the floor so you can trample on them. This is entropy in action, and that cannot be dismissed lightly. Chemicals don't want to be big complex molecules. Give them half a chance and they'll break down. So it's amazing that when we're talking about living organisms, they're managing to hold them together. Now, what is holding them together to become complex enough to form a living cell with the sort of complexity and the molecules that it needs? That becomes the big question. And all of this happening in defiance of entropy. So this is the backdrop for considering for our critique of the self-assembly theories of abiogenesis. Now the key thing to know is with abiogenesis, there is no standard model. There is no specific theory. In physics, we have a standard model for subatomic particles, and we have a standard model even for the Big Bang in the universe. We don't have a standard model for consciousness. We just have lots of different ideas, which none of them really are accepted or a coherent theory. Similarly, in abiogenesis, we have many, many, many theories, but nothing substantial, nothing that is accepted. So there's no theory of what even was the first life, never mind how that came about by self-assembly. But there's lots of efforts, and you've got to give credit for effort. So, when in trying to understand, well, where did the first life is, you've kind of got to think, well, what would that first life or living thing have been like from this particular perspective that you're adopting? So, many biochemists try to work backwards, and they try to conceive what might have been the first thing. Well, where are we now? Okay, so in the broadest sense, what we have now are three domains of life. There's bacteria, archaea, and eukarya. Now, originally, bacteria and archaea were considered to be practically the same thing, because when you look at them, they look very, very similar. You couldn't tell the difference from a diagram. But they rely on very, very different chemical systems. So they are so different in how they actually use energy and their internal processes that they have been considered separate domains of life. And besides these two, the prokaryotes, because they're single cell, they don't contain a nucleus for their genetic material. It just floats about inside them as single cells. So they're prokaryotes. However, there's another domain, the eukaryotes. And this is where the cells of these living things contain the genetic material kept wrapped up in the nucleus. Now, we got a little sign saying you are here, because the eukarya, these are all the different plants, all the different animals, all the insects, they're all wrapped up in this category. And human bodies are included amongst them. So, it's assumed that whatever existed way back gone separated at some point into two branches. One that produced bacteria, you see that little point of divergence, and the other started to form the archaea, and later on the eukarya. Now, therefore, an organism must have existed before that branching, and it is considered the common ancestor of bacteria, archaea, and eukarya. And it's the last one, i.e., it was the last thing that was the universal common ancestor. So it's that thing that must have existed which actually has given rise to every other type of life that we know of, whether bacteria, whether archaea, whether a eukaryote. So that's called LUCA, last universal common ancestor. However, before that point, there must have been some simpler first living thing that developed into LUCA. So now we have to consider, well, what is that thing that would have developed into this last universal common ancestor? And therefore LUCA was the descendant of that. So what was it like? Well, do we get any help from fossils, for instance? Well, the oldest life that scientists think that they have identified existing within the fossil record are the fossils of microorganisms that form a colony of stromatolites. These are structures which are created by stuff that is excreted by blue-green algae. And you find them in different places. Australia has, I think, the oldest, and they're dated back at least 3.5 billion years. Now, note that in the Atma paradigm, I'm going to be quoting dates as they are given in the standard literature. There may be quirks, there may be mistakes, there may be omissions. within the dating that is in the standard papers. But our thesis on consciousness-based life doesn't require us to challenge those dating methods. We're quite happy just to kind of use it and try and understand the principles of how consciousness works within living organisms. So stromatolites are therefore considered to be the oldest life that we have seen on Earth, which is interesting because they are living things. And in fact, this type of blue-green algae and the stromatolites are still existing today. Recently, they were discovered in my own home country in Northern Ireland at the Giant's Causeway in a little pool there. So they're not even simple enough to tell us what the first life could have been like. Even the simplest bacteria has 100 genes, and these must have been, first life would have to be simpler than that if it was going to be self-assembled. So perhaps, well, what's simpler than bacteria? Well, viruses, okay? And you've certainly heard a lot about viruses in recent times. They have fewer genes than bacteria, they're simple in structure, and therefore people think, well, maybe they were the first. But no biochemist will suggest that they are the precursor to life, because viruses cannot exist on their own. They require the existence of an actual living cell to invade and use for reproduction purposes. So they are not themselves a contender as the first form of life. So people have to, from all of this, we just have to use our imagination to conceive of the simplest living being. And it's reckoned that really you can boil a living system down to just 3 interacting systems. Note they say systems, not just ingredients, but actual systems. So the 3 systems are a membrane, a metabolism, and genetic material, genes. So you need structural integrity. You need an outer membrane that is going to hold it all together. And we'll see why that's important. You need metabolism, which is a set of chemical reactions that gets energy from their surroundings, from outside of the system. And these produce reactions which produce proteins and specialized proteins called enzymes in order to facilitate all types of reactions to carry out the functions that the cell needs in order to maintain its living status. And there's a third thing that's needed, reproduction. So this requires some genetic type of molecules. And it needs genes so that they have information for specifying the proteins and building the cells in the first place, but also that can be passed on to the next generation of cells. Because if they didn't create a next generation, then the cell ultimately would pass its sell-by date, disappear, and there would be no more life. So reproduction is required. And these are molecules of nucleic acids, such as DNA and RNA. Now, it's clear that these processes are absolutely intertwined. But perhaps that was a fact that was generally not realized, or perhaps put aside in the efforts to determine a path to life, practically to this day. But it seems in recent years, biologists are reconsidering the situation. So here's what they're coming up with. What comes first? Is it the membrane? Well, membranes are very simple to make, actually. They use a molecule called a lipid, which has it's the thing with the red head and the kind of yellowy orange tail. So the head is hydrophilic. It likes water. But the tail is hydrophobic. It doesn't like water. So they naturally line up in this double layer. The head that likes the water faces out. The tail that doesn't like water points in. And these kind of come together. And if you take lipids and you add them to salt and water and just shake them up, they will form these layers, and they will also form into spherical blobs, like in the picture. So this is very encouraging for the biochemists. They think, well, this is starting to become like a proto cell. We've got the membrane sorted. And fair enough, simplest of all the functions, but absolutely useless on its own, because lipids don't do anything else other than form this type of structure. They don't carry genes, they don't produce proteins, and they're not able to vary the way the membrane works. So you need something else. You're going to need proteins, right? Because proteins are, and proteins need amino acids. So some people think that proteins must have come first, because they're a very useful type of molecules and chemicals. They'll do a lot. And for proteins, we need amino acids. And for them, well, you need some carbon, nitrogen, oxygen, and hydrogen. Now, this was the challenge that Miller and Urey set themselves, how to produce some amino acids from the simplest substance that they could. So they chose ammonia, methane, and water vapor. Put them together, just heating, electricity, distillation, and bingo, they got a set of amino acids. At least a very thin soup of these, of 9 of the amino acids, particularly glycine and alanine, which are the smallest. No problem. You can create chemical reactions in the lab. The question is, can this be replicable in the natural situation? When you're looking at the questionable starting conditions, many people have said, well, did we really just have ammonia, water, and methane without any oxygen in the atmosphere at that time? The mood has changed on that one. An extremely low yield, but particularly an unstable product, because practically as soon as the amino acids are formed, they break down again. And the only way the experiment can do it is by whipping them away, isolating them, and not allowing them to interact with the rest of the reaction that's going on. The other problem with Miller's amino acids is that they're right-handed, and living systems on this planet only use left-handed amino acids. So none of the amino acids that are produced by this process actually are used in living organisms. So there's a lot of skepticism that this could happen, and particularly because they produce chemicals which stop the reaction progressing to the next stage. So the great thing about amino acids, though, is that they're able to form chains, and therefore they're considered the building blocks of life. So a little bit of chemistry about an amino acid. There are lots of different amino acids, but life tends to use 20 of them. And they vary according to the R thing. That's the side chain. You see circled in yellow. Okay, so that's the side chain. And that can be practically almost any other chemical. The thing that makes an amino acid is the N bit, or the 2 N bits. One, it has an amino group, which is an atom of nitrogen and 2 of hydrogen, and it's got a carboxyl, or the acid bit, which is carbon with oxygen and carbon with an oxide, OH connection. So that is the structure of an amino acid. And here's how they link together at the top of the picture there. The carboxyl, the COOH bit, reacts with the amino acid of the one next to it. So what basically happens is the carboxyl loses hydrogen atom, ion, and oxygen, and it takes a hydrogen one from the amino side of the other amino acid. That forms water, so you remove that, and the 2 amino acids link together, and that is called the peptide bond. Now, it's a dehydration reaction. It means it's getting rid of water, and it really needs to get rid of that water, because the peptide bond is now vulnerable to being attacked by the same water that was released. So the water produced by the reaction has to be removed. And therefore, if this is going to happen in nature, you have to have a situation where you might have water initially, but it needs to be a situation that can get rid of water, a drying circumstance. Or the formation of peptide bonds has to take place within a protected membrane, one capable of ejecting water, which is more complex than that simple spherical blob we just mentioned. So ignore all that. That's still, if we can get over that step, let's move on. Now, the situation of needing wetting and drying has given rise to suggestions that perhaps we need these amino acids to link together on a template, on a structure. So particularly maybe on something like silicate clays or some other inorganic chemicals might form a surface by which the amino acids could kind of sit and be attracted to, and perhaps even line them up in a particular way. So there's been some good progress on this, and particularly using what they call layered double hydroxides. And you see on the little diagram the little amino acids with the triangle and the two circles links up with the next amino acid with the square and the two circles, and then they link together. So this seems to produce what are called proto-biopolymers, and they produce peptides, not polypeptides, because they don't have that many, but oligopeptides, which has a few peptides linked together. But the yield, again, is very small. But people are trying to see how they can improve that, and it's found that using certain substances like structural copper, you can get a bigger yield, but only to produce bipeptides, only two amino acids linked together. So no matter how much we seem to try and how much this is touted as a process by which amino acids are coming together and starting to form polypeptides, and it's, you know, we're nearly there for life. No. Still got a long way, and it's considered that on their own, in the natural situation, we don't seem to be able to get very far with that. Proteinoids was another thing that were touted in the 50s and 60s, and the idea was that these are taking amino acids and trying to link them into chains and form what are called proteinoids, but they don't have any specific arrangement and they don't do anything. They can catalyze some reactions, but not do an awful lot. They're not life, they're not considered a protocell, they don't match or resemble the activities of any living being, and although they're continuing to hunt for life-like behavior in simple protein, that's still going on, but according to the recent review in the literature, the idea that protein started life has now been largely rejected. So the more recent common thought is to do with RNA. It's assumed that before you get to DNA, you might have RNA. After all, it's simpler than just simply because you only got one strand of the molecule rather than the two of DNA. So this is the idea of what's called the RNA world. RNA as the precursor to life. Somehow self-replicating RNA chains appeared on Earth, and they evolved or changed somehow into ribosomes. Now these ribosomes are like enzymes that can catalyze other chemical reactions and even make copies of themselves. So people think, well, this might have acted as the first proto-genetic code. So this was getting very exciting, and I think you'll probably find in most textbooks the RNA world is being cited as the way forward. This is where we're going to find the answer to the origin of life. However, the literature points out to key problems in this theory. It requires simultaneous self-assembly of both RNA and peptides in order to make it work, and there is no idea of the chemistry that must have occurred in order to produce the first strand of RNA. So we have no idea how to get to that RNA, even if you think that it can do something after that. However, even in practice, after struggling to get RNA to self-assemble or to copy itself in the lab, biochemists now concede that it needs a huge amount of help to do anything resembling that. Therefore, the hope of the RNA world is now considered, frankly, a dead issue. And this realization that there is no chance of getting anything that resembles a first living cell unless you have all the features in place at once. You need the proteins, you need the genes, you need the membrane. They must all appear together. They must be ready to maintain the key processes of living organisms, the metabolic reactions that are going on inside the cell. So metabolism involves creating entire sequences of interdependent chemical reactions controlled by a large number of proteins and enzymes. And these are proteins and enzymes which didn't exist before the first cell appeared. Why not? Because of the chicken and egg problem. You see, enzymes are controlled proteins. Proteins need enzymes to form and to function, but enzymes are a particular type of protein. So which came first, proteins or enzymes? Chicken and egg. Okay? And the more you look into the complexity of this relationship of all the chemistry related to the formation and functioning of proteins and into DNA and RNA, the more there seems to be this type of chicken and egg issues. For instance, proteins and DNA. DNA manufactures proteins, but proteins are needed to make DNA. Which came first? Chicken and egg. You know? Or do they have to both appear simultaneously? Therefore, one cannot have produced the other. They all need to be self-assembled. It's not that you can assemble one thing and it produces the other. Similarly, with the DNA and the membrane, another chicken and egg. The DNA can't survive, even do its job outside of a fully functioning cell. It requires the protection of a fully operational cell to exist and to function. But a cell membrane that can sustain the processes that enable DNA to replicate isn't just a simple blob that we saw earlier. It requires to have particular features, and so that it can allow things through and accept in and expel things out. It needs to have particular features in order to operate and to form in that way. And guess what? That requires the specification of proteins produced by DNA. So which came first? Now, in considering these issues, it's important to appreciate the inherent complexity of any living cell, even the one we might imagine to be that first, simplest possible thing. Because I think we're still stuck in the 19th century. You know, this is the idea that you can kind of think of the cell as a sort of blob, soft, mushy stuff, the sort of thing you might find in a stagnant pool of slime. But it's not. Even the simplest cell must be a complex set of interacting functions, each reliant on the other parts of the system. There's feedback loops, signaling pathways, there's ways in which they control the information being used within the cell, and also how that information is shared with other cells. There's fine-tuning of all the different features and processes. There's even error correction systems. Now, Michael Katz in his book argues that cells and organisms are built on heterogeneous elements arranged in heterogeneous configurations, and they do not self-assemble. What he means by heterogeneous is that the systems, the information and the organization of the operating whole cannot be broken down into constituent parts, because each is so interdependent and based on the relationship it has with the other elements for its function and for its existence. You cannot assemble that without a chicken and egg problem. He says you can't stir together the parts of a cell and assemble a neuron or a walrus to create. He says to create a cell or an organism, one needs to have a pre-existing cell or organism. So the simultaneous presence of pretty well every feature of the cell it produces a multitude of chickens and eggs. And this is why recently there's been recognition that all the elements that are needed for a living cell must come together at exactly the same time in exactly the right circumstances to have the first functioning living cell. Now, this means that biochemists are conceding that the previous ideas that have been peddled of the gradual build-up of one bit after another, invoking the power of evolution where it shouldn't be invoked, is problematic. And it offers little hope of ever being a coherent theory. So what these biochemists are exploring is what's called everything first theory. Okay? And they admit that actually the idea that everything can come together all at once, all together, all ready to go, means that you need a sort of Goldilocks chemistry, and that's their term, not mine. Having a mixture with enough variety for complex reactions to occur, but not so much that it becomes a jumbled mess. But huge questions still remain. What are the components in the systems related to metabolism? Where does that come from? They seem to be able to, say, well, we can maybe get the membrane sorted, maybe we can get some protein type things. But what is the system that produces all these processes all together? No one has figured that out, or how, when, or where it could have happened. So the everything first theory is not an answer to the start of life. It ups the odds. It increases the astronomical probability, that life, the living entity, living thing, has to overcome in order to have happened according to chance self-assembly. So we're back to probability and chance again. The idea that somehow or other we have these crazy odds of all of this happening, not by the process of, well, we can show how this happens and this and then this and this. No. That the idea that all of this can come together in one go, that's the odds we're now having to deal with. Now, worth just clarifying probability and chance. Probability is to do with statistics. It assesses the odds of a particular outcome occurring. Now, there's two ways of really establishing odds. One is you can figure it out related to the event and how the event is occurring. For instance, coin tossing. By the nature of tossing a coin, it's going to come up heads or tails. And by the nature of the physics of the coin and the way it's tossed, you expect it to come 50-50 roughly after a time. It'll vary a little bit, but you can kind of put rough odds to it. Or another way that you can assess probability is to do it after the fact and see how that might apply to future events. For instance, using the coronavirus as our example, we might say how many cases of COVID-19 were diagnosed last week, okay? And then we might say, well, okay, what are the odds of infection rising in the population? So from our statistics that we have measured in the past, we might be able to create the odds of infection. We're not able to look to produce the statistics from just looking at the starting points. We have to kind of have measured them in the past. And that's the difficulty that our scientists are having with coronavirus at the moment. They need basically the coronavirus to have happened, to have gone through the population, to have amassed all the data around the world, and then say, here's how this epidemic works. It's very hard to do it as a predictive thing. Now, neither of those processes is particularly useful in figuring out the odds for life arising from self-assembly. We haven't got many incidents of life appearing. We're not even sure we got one, but we're not going to see it again. So, and we don't have a system, so we don't have a system for looking at past data, nor can we figure out the odds from the the properties of the thing we're dealing with. We're not dealing with just a simple coin toss. For instance, with RNA, which is described as the prebiotic chemist's nightmare by Dr. King Smith, it requires 14 major hurdles with 10 steps each. And some people try and work out a probability of this, and it's something like 10 to the minus 109 so that's 1 against 10 with 109 zeros after it against its successful formulation. Now, I personally don't think we should rely on any type of probability odds that are quoted for or against the self-assembly of life. All I think you can say is the odds are really, really, really big. Now, scientific evidence, and this is the key point, science is meant to establish evidence to a very high degree of certainty. And really that certainty should be 4.3 million to 1 in favor of something being established as, you know, as good close to a scientific fact as you can get. So it's 4.3 million to 1 in favor rather than millions and millions against. So why are we being told that this is the scientific approach to the formation of life? That's what really concerns me. Now, because whenever you're dealing with odds, whether the odds are big or small, it's still you're believing in the efficacy or the ability of chance to have been the cause. So whatever those odds are, however big or however medium you might think that they are, or even however likely, abiogenesis relies on the belief, your belief in the efficacy of chance. Now, the whole exercise of prebiotic chemistry is trying to reduce these astronomical odds against self-assembly and trying to bring them into the realm of something more believable. But that doesn't change the principle that at the heart of this thesis of abiogenesis and self-assembly lies the non-scientific postulation that there is no specific cause at work, just the chance interactions of components acting in terms of their pre-existing properties, which is very limited, because it's relying on two low information aspects. It's relying on the limited self-organizational physical and chemical properties of the components, which can do something for connecting together, but not a lot, and two, their chance interactions as assisted by the environmental circumstances. Now, if you postulate the creation of high information system, like even the simplest living thing, from these low information processes, and you're not able to show any account of how this could have come, other than it seems that we believe it happened by chance, fine. Believe that, but please do not call this a scientific proposition. Choose to believe it and promote it as your own unproven blind faith, but it is not science. Now, some people and some chemists try to bring in the opportunity that time offers. As Francis Crick once said, the origin of life appears to be such almost a miracle. So many are the conditions that had to be satisfied to get it going. But he says, this might be overcome in long periods of time. So the idea is that over a long period of time, that which seems an impossibility becomes a certainty. And this is summed up in the thought experiment of the infinite monkey cage. The idea that you've got an infinite, not just a lot, an infinite number of monkeys on their typewriters or their keypads, and they're battering away there. Sooner or later, one of them is bound, absolutely bound to type out all of Shakespeare's sonnets, or some say his complete works. It's just a matter of running the odds for long enough. Okay, but this is a question of bringing in eternity and infinity into the picture. It's not science. If, you know, you can say, well, not just a living, why just a living cell can be produced? You can produce unicorns and Santa Claus and all the other things that you don't believe in. Now, The big question is, though, if you're going to quote time as on your side, do you really have eternity to play with? Do you have an infinite number of years? No. Here's the Earth's timeline in the simplest format, right? The evidence, I mentioned about 3.5. Recently, there's aspects which seem to take that back to 3.85 billion years ago. Now, we're really just talking about the period when the Earth had just about sorted itself out from its Hadean state, and it was still going through this fiery process. And it means that abiogenesis must have taken place either in the sea, close to 4.4 billion years ago, or on land, which would have had to have been after 4 billion years. So you actually see there isn't a lot of time to produce the first cell. At the very best, maybe 500 million years. That sounds a long time, but in quoting eternity and infinity and all your monkeys, that's not a lot of time. And you don't want to be around on the Earth at that time. It is not a very healthy place for life. So where did life actually happen? Well, again, there's so many contenders for this. Is it in the sea? Is it in hydrothermal vents? Is it on the beach? Is it a pond on land? Is it under ice? Is it volcanic geothermal pools? Is it rocks being dried and wetted? And it's really confusing, because for many scientists, any idea of talking about water is a non-starter. Life likes water when it's living, but the chemical processes that seem to be needed for self-assembly do not like water. So that becomes the paradox. And of course then people have suggested, well, maybe the first life just comes from outer space. Somehow it arrived here on a meteorite. And that's still talked about as a possibility. And I think this is just hedging bets. Why? Because people know that suggesting that it came from elsewhere means you've still got the same problem of trying to define how that living thing came about by self-assembly elsewhere. And if you can't figure out the situation on Earth to get going, how are you going to give us a scientific description of how it happened elsewhere? And also, biochemists explain that panspermia, this idea of cells coming from outer space, cannot be true. Otherwise, bacteria would be raining down on us from space, and it's not. Neighbouring planets would be scattered with their mates. They're not. There's no indication of incoming bacteria. And even the experiments done outside the International Space Center station show that space is not kind to life. Therefore, any living thing that is travelling on a meteorite would not survive very long. So where is it coming from? Within our solar system or beyond? In talking about the synthesis of life, it's important we mention Craig Venter, who was publicized as having synthesized life. He basically created a synthetic genome of 1 million base pairs. And that's a huge engineering technological challenge. He worked out the exact code of a particular organism, the genetic code. He stitched together all the base pairs, and then he took that DNA and inserted it into a living cell. So no one can suggest this is creating life. All he did was create a synthetic copy of DNA. And that's fine. DNA is simply a chemical. It's not a magical formula. It's not a magical thing that somehow or other activates life. No, it is simply a chemical. And we'll talk about the nature of DNA in later discussions. So we don't accept that as, it's not a synthesis of life. He is using DNA, which is copied from a living organism, and he's just popping the synthetic chemical back in, and it does the same job as the naturally occurring one. No problem, no issue whatsoever. So to conclude this explanation of life arising by self-assembly, I quote Dr. Yockey here from UC Berkeley. You must conclude that no valid scientific explanation of life exists at present. Since science has not the vaguest idea of how life originated on Earth, it would be honest to admit this to students, the agencies funding research, and the public. I doubt that's going to happen. So just to be clear, there's no standard model, not even a consistent batch of ideas, no clarity on what the first living thing was, other than it needed the three components. There's no theory for self-assembly of these components, other than the idea that they now have that they need to reassess all the other theories they've had so that maybe it can happen everything first or all at once. There's no idea of where, when it could have happened. So after all the efforts to try to reduce the unlikeliness of self-assembly, the odds for it happening, I think, seem to have been lengthened by the everything first idea. And the short time available on Earth, and that time seems to be getting narrower and narrower, is not helpful to leaving things to chance. And the idea of life arriving from space, now pretty well discounted. But still, it's an option for you to believe in if you wish. I'm going to skip the next couple of slides and just introduce where we will be going for next week. What we're going to be suggesting is that we look at life as something that is based on consciousness. Where does all this information come from that is needed for the specification of any living being? Well, we have a source of information. That source is consciousness. And we have shown that consciousness has the ability to organize physical matter. Could this have a bearing on biology? Could this have a bearing on how life appears fully formed? Let's see that in the next few talks here at the Atma paradigm. But I apologize. We have run over our hour, and if there's any questions and answers, I'll try and deal with those by email. But we're not able to take any further at this time. Apologize for that, but I hope that you'll be with us next week when we'll be starting to look at life forms, the actual bodies and creatures that make up this living world. And we'll be looking at the issues of speciation and evolutionary theory. So that's next Thursday, the 3rd of September, UK time, 6 o'clock. Hope to see you then. Thank you very much. Hare Krishna.